So hi everybody. For those of you that don't know me, my name's Dr. Jason Brown, and I've been asked to deliver a vaccines lecture to two cohorts. The first cohort is BDS3, and the second cohort is to the oral science master's students. So if we make a start, these are the intended learning outcomes. So Professor Ramage has provided me with the ILOs for the BDS3 cohort, and specifically these two here. So these are should be covered in to some degree within this vaccine lecture, specifically when we talk about infection control measures. And of course, vaccinations are excellent infection control measures. In terms of the oral sciences master's cohort, Dr. Fortuna has provided me with or has told me that you should be able to find the ILOs on the Moodle website, and specifically this vaccines lecture is part of this module, and of course it kind of covers the therapeutics part of this module. So if we make a start on the contents for today's lecture, these are documented in this slide here. So what we're going to do is talk about vaccines. So what are they? How do they work? I'll provide to you different types of vaccines, specifically provide some examples of vaccines, and given the current situation in the world, of course, we would have to talk about COVID vaccines. So I'll provide to you the two types of COVID vaccines that are now being rolled out within the UK, and also the biological mechanisms behind them. We'll then talk about vaccine efficacy, so this idea of how scientists can test how effective a certain vaccine is. And then we'll cover the principles of herd immunity. So this is a term you've probably encountered or heard within the news within the last 6 to 12 months or so. And then finally, we'll talk about problems associated with creating vaccines, and specifically talking about viruses and the mutations that can arise within those viruses that can actually make creating vaccines quite difficult. So firstly, if we make a start, so the first question that we want to ask is, what are vaccines? So the word vaccine was first coined in 1798. So you may or you may not have heard of Edward Jenner. So he was the first to demonstrate in the late 18th century that this virus here, Varicella vaccinae, which is ultimately the cause of cowpox, was able to be used to prevent smallpox in humans. And this is highlighted quite nicely in this YouTube video. What I do is I tend to not play the YouTube videos in my sessions simply because it seems to always crash my Zoom recording. So what I would recommend is watching the YouTube videos when you are rewatching this presentation. So it was first coined in the late 18th century, and vaccines apply to ultimately all biological preparations that are produced from living organisms that are injected or delivered, administered into humans that can enhance immunity against disease. So there's two different types of vaccines. So we have prophylactic vaccines. So these help to prevent disease. So COVID is a good example. And then we also can have therapeutic vaccines as well. So these are used to treat diseases. And generally speaking, vaccines are administered via injection, but they can also be administered via oral and intranasal routes as well. So the next few slides are just to emphasize how important vaccines have been within the last century. And you can see here on the right-hand side, this figure highlighting all of the diseases that have now essentially been abolished, or have at least been severely reduced within the human population, simply because of the production of vaccines. So if we go back 100 years, you have things like TB, diphtheria, typhoid, measles, mumps, rubella, hepatitis, moving through to the current day. And if you can recall, in the late 2000s, the swine flu pandemic that hit the world, there is now a vaccine for that. In recent years, we've obviously had an outbreak of Ebola, or outbreaks of Ebola within specifically within African countries, but also in people that have spread it into other countries. And then, of course, last year, end of 2019, was COVID-19, and that appeared specifically in China, and then, of course, spread across the world. And sort of towards the end of last summer, and moving it through to the autumn and the winter, there was a vaccine or different vaccines that were created for COVID. And these are the 4 that have passed clinical trials and are now within circulation within the world. And the 2 that we're most interested in, or at least the 2 that you've specifically heard of within the context of at least the UK, is the Oxford AstraZeneca virus vaccine and also the Pfizer BioNTech vaccine. And we'll talk about the mechanisms of these later on in the session. But the take-home message here really is that vaccines work. So if any of you do know any anti-vaxxers out there, I would highly recommend showing them this slide, that vaccines do work against many different infectious agents. So the current vaccines or vaccinations that are in circulation within the world are listed here. These are a list of them. There's many others as well, and you can find these at the World Health Organization website. But vaccinations can generally be developed against most infectious agents. But as you can see from this list, most of these infectious agents are either bacteria or viruses. So currently there are a number of fungal vaccinations that are in clinical trials, but none have passed all of those clinical trials and none are within circulation. And there are also other organisms, other microbes that you cannot really create vaccines against. So things like parasites. Parasites have very unique life cycles, so it's very difficult to create vaccines against them. And there are also other complications and other anomalies can exist when you are creating vaccines. So certain organisms that have high mutation rates, things like viruses specifically, for example, the high mutation rate in certain viral families can, of course, affect the production of vaccines. Certain organisms that have specific immune evasive mechanisms, quite difficult to create vaccines against these, simply because the microorganism does not elicit any sort of immune response. And as we'll come on to talk about, the whole importance and process of vaccinations involves the immune system. We've talked about the parasites and, of course, new and emerging pathogens. So certain organisms that we don't know too much about, it's very difficult to create vaccines against them, at least efficiently, and of course in a short space of time. So things like Ebola and coronavirus in recent years are great examples of these. But the take-home message here is that the vast majority of vaccinations that are within circulation are specifically for bacteria and viruses. So we're going to move on to talk about how vaccines work, and specifically I'm going to talk about the role of the immune system and essentially what is happening within the immune system when you are vaccinated. So I hope for the vast majority of you, this will be a recap of things that you were taught in the early years of your BDS course. For the BDS3 cohort, this was probably delivered by Dr. Nile a few years ago, and this is essentially what I teach now for the new BDS1 cohort. For the oral sciences students, I hope this is something that you were taught in your course. If not, we can try and go into a bit more detail in the live session on anything that maybe doesn't make sense. So if we make a start, so how do vaccines work? Well, ultimately, the primary aim of vaccines is to provide and create immunological memory. So in individuals that do not get vaccinated and those that are infected with a certain disease-causing agent, then basically this disease-causing agent can cause infection and drive an immune response. And the immune response will lead to some level of immunological memory in certain patients, and in the vast majority of patients, they will recover from the disease. However, of course, if they've not been vaccinated, then in certain individuals, death and disability may arise. In the population that have received the vaccine, this involves injecting an antigen, and we'll talk about antigens in greater detail in the coming slides, injection of an antigen into the host, into the human body, there is no disease, but there is an immune response to that antigen. And this immune response will lead to immunological memory. And this will basically prevent, or at least improve how the immune system responds to that disease-causing agent if it infects you at a later date. So vaccines, the process of vaccination involves both the innate and the adaptive branches of the immune system. So this is a very simple way of looking at things here, and we'll go into a bit more detail in the coming slides. You have a disease-causing agent that will be recognized by the immune system. The immune system, the innate and the adaptive immune system, will work together to basically drive an antibody response. This antibody will then be able to target that disease-causing agent and drive destruction of it. Within the context of vaccinations, you are vaccinated with a certain antigen against or of a certain microbe. This antigen will drive an immune response. The immune response will ultimately then result in the production of antibodies and memory B or T cells. And these memory B cells or T cells will basically drive a more effective immune response against that microbe if it infects you at a later date. But ultimately, the primary mechanisms in this process is the same if you are infected with a disease-causing agent or if you were vaccinated against it. It's basically the immune system responding to it, recognizing it, and targeting it for removal from the host. So if we move on and talk about specifics, so there's two branches of the immune system. So we have the innate immune system, which is the first line of defense. So this is non-specific. The innate immune system will respond to any microbe that is invading the host. The adaptive immune response is more specific and acquired. And each of these branches can be broken down into different components. So you have cellular components. So this is essentially all of your different innate and adaptive immune cells. You also have humoral components, and specifically these would be your proteins that are produced by each of your innate and adaptive immune cells, so things like cytokines and chemokines, but also your antibodies as well. And the antibodies are specifically produced by adaptive immune cells known as B cells. So the innate immune response is very rapid. So this can occur within hours. The adaptive immune response is very slow. And this can occur between days and weeks. But the primary aim of the innate immune system, as I've said, is to ultimately respond fast to the microbial threat and essentially remove it before it can cause infection. The primary role of the adaptive immune system is to generate immunological memory. And there's basically interactions between innate immune cells and adaptive immune cells, which results in this formation of immunological memory within the adaptive immune system. And we're going to talk about the specifics and the specific mechanisms that arise during this process of vaccination. And so I've mentioned it a few slides ago. This process arises whether you're infected with a disease-causing agent or whether you are being vaccinated against it. The processes are very similar. So if we start and talk about the innate immune system and the innate immune cells that are part of that branch of the immune system. So innate immune cells, their primary aim is to recognize antigens. So antigens are ultimately just components of microbes. So these can be things like cell wall components or genetic material, for example. And the primary aim of innate immune cells is to recognize these using specific receptors. These receptors are known as pattern recognition receptors. These innate immune cells are found patrolling the tissue or the mucosal barrier. So if we're talking about vaccinations, the main type of vaccination or the main way that a vaccine is administered is via the skin, via an injection. So these cells are found patrolling the epithelium of the skin. But they're also found at other mucosal and barrier sites, such as the oral mucosa, as we said, the skin, the GI tract, and also things like the lining of the lungs. The innate immune cells' role is, and specifically dendritic cells and macrophages, their main role is to phagocytose and present antigen. So the process of phagocytosis is something we'll talk about in the next slide, which is basically engulfing the antigen, the microbe, degrading it, and then presenting it to a T cell or B cell. And this is essentially the important role of innate immune cells, is to activate T cells and B cells. So this is the process of phagocytosis, and essentially what happens is you have a microbe, or components of a microbe will be engulfed, phagocytosed by a dendritic cell or a macrophage, or the umbrella term for these is an antigen presenting cell. And basically what will happen is they will process these microbes, process these antigens by degrading them intracellularly. And they do this by forming certain vesicles within the cell itself and driving lysis using digestive enzymes of these microbes, and then either releasing them or presenting them to other cells. And this would be then the process of antigen presentation. So you have phagocytosis of the microbe, degradation of the microbe, processing of the antigen, and the antigen will then be attached and will form a complex with the MHC receptor. So the MHC is the receptor responsible for presenting the antigen to T cells. So basically this complex will be formed within the cell, within the cytoplasm of the cell, and the complex will then be delivered to the cell surface. At the cell surface, what happens there is that you get presentation of the antigen by the antigen presenting cell, such as the dendritic cell, which will then, via interactions with T cells and T cell receptors specifically, will drive the activation and differentiation of a naive T cell. And we'll talk about this later on in the session. So what you need to be aware of is that you have the process of phagocytosis of an antigen. So this antigen would be part of the vaccine formulation. You get processing of that antigen within innate immune cells, such as macrophages or dendritic cells. That antigen is then presented to the adaptive immune system to drive immunological memory. And the cells that are important in driving this immunological memory are B cells and T cells. So these are essential for either prolonged or recurrent infections, and specifically within the case of vaccines, are essential for providing you with this immunological memory in case that microbe at a later date will infect you. So your B cells are essentially a part of your humoral response. T cells are essentially part of your cell-mediated response. So B cells also have the potential to process and present antigen, and they'll do this to T cells. So they're actually termed antigen presenting cells in a very similar manner to certain innate immune cells. And we'll talk about this interaction between B cells and T cells later on. But their primary function is to produce antibodies. And these antibodies have the potential to basically attack and eliminate microbes. T cells, on the other hand, their important role is in cell-mediated responses, specifically communicating with different cells. And we'll talk about the different types of T cells in the next slide. So these are the different types. So we have T helper cells. So these function to help support other immune cells to fight threats. Cytotoxic T cells, they can destroy own cells, and specifically this is important if the cell is infected with an intracellular bacteria or even a virus. Regulatory T cells, also known as Tregs, these regulate or suppress other cells in the immune system. And then what you need to be aware of is that all cells start as a naive T cell, and they have receptors that are very specific for a given antigen. So they can change the structure of their receptor depending on the antigen that they want to interact with. And these receptors are known as TCRs, or T cell receptors. And it's these T cell receptors that recognize antigen that is presented to them by dendritic cells or macrophages. And the main cell type that we're going to talk about in today's session is T helper cells. I want to just make it clear that you need to be aware that cytotoxic T cells and Tregs are also really important in immune systems or in the immune system. However, I've only got an hour to talk to you about vaccines and the basic principles of vaccines. So with the interest of time in mind, I'm only just going to talk about T helper cells, but these other cells of the T cell lineage also have important functions, and it's important not to forget that they have these important functions. So how do T cells become activated by dendritic cells? Well, this is what happens at the skin barrier when you are vaccinated with a certain antigen. So dendritic cells, or antigen-presenting cells in general, will take up and process antigen in the epidermis. So this is at the injection site. Once they've taken up the antigen, so once they've phagocytosed the antigen, they will migrate to the lymph nodes, and they'll migrate to these lymph nodes, which are found somewhere within the human body. So for instance, lymph nodes, if you are injected within the arm, these lymph nodes, these draining lymph nodes would be found in the armpit. And these lymph nodes, they kind of look like kidney beans, have certain regions where the dendritic cells will interact with the T cells. So the dendritic cells will interact with the T cells within the T cell area of the lymph node. And you can see this from the fluorescently stained image here. So this is a lymph node, fluorescently stained with different fluorescent dyes or fluorescent probes. And you can see the dendritic cells are stained in green, whereas the T cells are stained in red. And what you're able to see is that there's interactions between the dendritic cells and the T cells. And ultimately what's happening here is you have dendritic cells are essentially priming naive T cells. And that is the process of activating and differentiating naive T cells into different subsets, such as T helper cells. And it's this process of priming is the scientific name for that. And this process of T cell priming, as we've said, occurs in certain regions of the lymph node. So you need to be aware that CD4 T cells, so we're talking about T helper cells here, they start life as a naive T cell. And ultimately, there's 3 signals required for the activation and determination of the fate of those T cells. And these are the 3 signals here. So once you have a dendritic cell that's phagocytosed, process the antigen, and then presenting that antigen to the T cell within the lymph node, you have 3 signals. So the first signal is signal 1, and this is the specific recognition of that antigen by the T cell receptor. Signal 2 is where you have co-stimulatory molecules essentially interacting with their specific receptors on their other cell type. But basically what you'll have is the dendritic cell will have certain proteins or receptors on their cell surface, and they will recognize certain proteins / receptors that are found on the T cell. And basically interaction between these receptors will lead to the second signal. And this second signal is just as important as the first signal in driving this activation and differentiation of T cells. And then finally, the third signal is the dendritic cell will produce certain proteins, depending on the antigen that is presenting, will produce certain proteins known as cytokines, which will drive the T cells or instruct the T cells to differentiate into different subsets. And when we're talking about T helper cells and specifically CD4 T cells, these can differentiate into different subsets. So T helper 1 cells, T helper 2 cells, and T helper 17 cells are just some good examples here. And what I want to make clear here is that T helper 2 cells specifically are really important in interacting with B cells. And it's the interaction between these T cells and the B cells which result in the production of antibodies to a given antigen that you have just been vaccinated with. So what are B cells? As I've said, they communicate with T cells and specifically T helper 2 cells. They have specific receptors for antigens. These receptors are essentially antibodies. So they have antibodies that are found on the cell surface of the B cell, but they can also produce antibodies which will be released by the cell. And we'll talk about these in the coming slides. And basically what happens is B cells have the potential to be an antigen presenting cell, so they can actually phagocytose and process antigen in a similar manner to dendritic cells, and they will then interact with T cells via this antigen presentation process. And following this interaction, which we'll talk about in the next slide, you will get the mature B cells would differentiate into either plasma cells. So plasma cells are great big antibody factories. So they just produce copious amounts of antibody which can target a certain microbe or a certain antigen. They can also differentiate into memory B cells. So memory B cells are really important in mounting a quicker antibody response if you have a subsequent infection by a given microbe. And we'll talk about these two types in the coming slides. So B cell activation arises either with or without the help of T cells. And these are the technical names for this. So antigens, which can drive the activation of B cells without the help of T cells, are known as thymus-independent antigens. Those antigens that require T cell help are known as thymus-dependent antigens. Okay, and the ones that we're specifically interested in when we're talking about the process of vaccinations is this first one here. So the interactions of B cells with T cells will result in the generation of plasma cells, but also memory B cells. And basically what happens in the process here is you get receptors on the B cell, which are essentially a certain type of antibody, will recognize the antigen. They'll phagocytose and process that antigen, and then they will present that antigen to T helper cells. And there'll be T helper cells that have already been interacted with dendritic cells, so they already know the structure of the antigen. So they're able to recognize that antigen and basically tell the B cell via the production of cytokines to either become plasma cells or memory B cells. And this is also shown here in this diagram here, where you have phagocytosis of the antigen, processing of the antigen by the B cell, presentation to the T cell. The T cell will then produce certain cytokines, which will instruct the B cell to proliferate into memory B cells or plasma cells. So there are other interactions. So there are co-stimulatory receptors that drive this process, but it's ultimately the cytokine signal that's released by the T helper cell, which will induce proliferation of the B cells. So just a comment I want to make here is that when we think about the other process, so the thymus-independent antigen process, you don't get the production of memory B cells. You only get the production of plasma cells. And this is, of course, not important or not as important when you're talking about the process of vaccinations, because you don't get any immunological memory when you produce plasma cells. It is the memory B cells which are really important in basically driving an immune response if you are infected at a later date with a given organism that you have you've been vaccinated against. So if we just move on and talk a bit about the antibody production by B cells. So B cells produce 5 different types of antibodies. So these are the 5 here. The 2 that we're most specifically interested in are IgG and IgM within the context of vaccinations. There are other types, so IgE, IgD, and IgA have other functions within the human body. So IgA you're probably aware of because it's found in saliva. IgD is essentially the receptor that's found on B cells. So this receptor is what will recognize certain antigens before the process of phagocytosis. IgE is important in hypersensitivity reactions and in allergies. But the 2 we're most interested in here within the context of vaccinations are IgG and IgM. So activation of B cells will lead to a process known as class switching. And I appreciate you might not know what class switching is, so we're going to describe what it is in this slide. So IgM, which is this antibody here, this is the first antibody that is produced by an activated B cell. However, this antibody has a quite low affinity for a given antigen, and therefore the B cell needs to class switch into producing IgG, and this is what IgG looks like here. And the importance of this class switching is emphasized by the idea of affinity. So IgG has a higher affinity for a given antigen, and therefore it can drive a bigger and a better immune response against a given antigen, whereas IgM has a weaker affinity and therefore its response is a lot weaker to a given antigen. And that is essentially the take-home message, is that IgM is the first antibody that is produced by B cells, but these IgM producing B cells need to class switch into producing IgG. And they do this class switching process because IgG has a higher affinity for a given antigen. And this hopefully should make a bit more sense when we talk about this figure here, and specifically when we talk about the principle of vaccination. So the primary response within the immune system can take several days to develop, and specifically within the adaptive immune system, it can take several days. This process of B cell class switching is really important to produce elevated amounts of IgG, and this is because of the idea of affinity, as we've talked about in the last slide. And what happens here is that you have the primary antigen challenge. So this is your first, essentially the first jab that you'll receive during the vaccination process. And then you have the innate immune system essentially responding to that antigen, processing the antigen, presenting the antigen to the adaptive immune response. And ultimately, this is the adaptive immune response here. So from about 3 to 4 days up to a few weeks, your adaptive immune system is taken over. During this process, the B cells will produce IgM. As I've said, IgM is the first antibodies that is produced by B cells. But because IgM has quite a low affinity or weak affinity for a given antigen, the B cells need to class switch to IgG. And this is really important because IgG has a stronger affinity and therefore can drive more of an effective immune response against the given antigen. So if you go and move on for 4 weeks to 5 weeks after the primary antigen challenge, this secondary antigen challenge can either be a booster, okay, so the booster that you may have to receive for certain vaccines, or it actually may be you becoming infected with that certain microbial infectious agent. And basically, following secondary antigen challenge, because your immune system has developed immunological memory, specifically IgG, that IgG antibodies that are specific for a given antigen, the production of IgG will basically drive a more effective immune response against that antigen. And it's because of this is why the process of vaccination is so important, because when you have been vaccinated against a certain antigen, IgG antibodies will be produced at a much greater and much faster rate following secondary exposure. And it's essentially IgG which is important in driving a more effective immune response. So I appreciate what I've talked about may be quite overwhelming for you as dental students. I do appreciate that it is quite intense immunology in terms of topics. Hopefully this overview will kind of give you at least an idea of what I've just waffled on about for the last 20 minutes. But basically, you will be vaccinated with a given microbe, so a bacterial or viral antigen. So this is the skin epithelial barrier. You have innate immune cells and also B cells and T cells patrolling the skin epithelial barrier. First thing that happens is you get recognition of the microbes. So the immune cells via certain receptors known as pattern recognition receptors will recognize the antigen. That antigen will then be phagocytosed and presented to T cells. So this process here is occurring at the epithelial barrier. This process here is occurring within the lymph node. And basically what happens is you have presentation of the antigen by the antigen presenting cell, such as the dendritic cell, to a naive T cell. And this process happens in the draining lymph nodes. And then basically you have three signals. So the receptors and the co-stimulatory receptors on the two cell types will interact. Production of cytokines by dendritic cells will instruct the naive T cell to differentiate. Differentiation of the naive T cell into T helper subsets, and specifically T helper 2 subsets, will also play a role in B cell activation. So T cells will interact with B cells, and the T cells will essentially instruct the B cell to produce antibodies against a given antigen. And essentially it's these antibodies which provide to you this immunological memory against a certain antigen or a certain microbe that you may be reinfected with, or you may be infected with following the vaccination process. And it's ultimately these antibodies, and specifically IgG antibodies, which give you this prolonged immunity against a certain antigen. So it's a very, very simplified way of looking at things, but hopefully it gives you an overview of what I've just talked about for the last 20 minutes. So if we swiftly move away from the immunology side of things, and I'm going to talk about in the next 20 minutes or so, the different types of vaccines and also some terms that you may encounter when thinking about vaccines. There's different types of vaccines, so these can be composed of different components of infectious agents. So on the left-hand side of this slide, you can see the more commonly used examples of vaccines by type, and we'll talk about each of these specific ones in the coming slides. On the right-hand side, you have what are known as next-generation platforms. So essentially, the new methodologies that are being used to try and counteract these rapidly mutating viral infections or viral viruses. And specifically when we're talking about the COVID-19 virus, the viral vector and the RNA viruses are specifically what are being rolled out within the UK at the time being. And we'll talk about these and the mechanisms for these in the coming slides. So if we start and talk about the more classical types of vaccines, so we have the live attenuated vaccine. So this basically involves taking a viable microbe and in some way attenuating it. And this can be by basically growing the organism in suboptimal conditions. So the organism is still viable, and because it is still viable, you likely only need 1 or 2 doses of most live vaccines to give you a lifetime of protection. So it's arguably the strongest type of vaccine that you can receive. So a good example would be measles, mumps, and rubella vaccines, a vaccination that you received at a very young age, and then you probably may need a booster 20, 30 years after your initial vaccination. And ultimately, this is a viable microorganism that is being injected into the human body. And it's essentially used for certain diseases. The second type is an activated vaccine. So this would be taking the disease-causing agent and basically inactivating it in a certain way. So this can be either thermal or chemical treatment of the microbe, which will basically kill the microbe. And then following the purification step and the formulation of the vaccine, this will then be injected into you. And a good example of an inactivated vaccine would be influenza. Because it's inactivated, because the organism isn't viable, you will likely need several doses of these. So you'll need boosters throughout your lifetime. The other types are the recombinant subunit vaccines. So this is where scientists will identify a certain gene which is responsible for transcribing a certain protein. And this protein is essentially expressed in at a high level in a cell type within the laboratory. And basically these proteins are purified, so you have a large quantity of them, and this is then formulated into the vaccine. And recombinant vaccines, again, you will likely need boosters. Good examples of these types of vaccines would be hepatitis B. The fourth type of vaccine would be toxoid vaccines. So this is where you would purify, or scientists would purify, a certain toxin that is produced by a disease-causing agent. This toxin would be inactivated via, let's say, chemical treatment. The inactivated toxin would then basically be formulated into the vaccine, which will then be delivered and administered. And a good example of toxoid vaccines would be tetanus or diphtheria. So what you need to be aware of here is the idea that it doesn't have to be the microbe itself, it just has to be an antigen of a given microbe. So this could be a toxin, this could be a component of the cell wall, component of the genetic material. And then the final type of what we would call a classical vaccine is the conjugated polysaccharide protein vaccine. And this is ultimately conjugation of sugars, polysaccharides, and proteins from different microbes to ultimately increase the effectiveness of a polysaccharide vaccine. And the simple reason for this is that sugars are they don't drive the same immune response as proteins do. So the immune system will recognize sugars from a given microbe, but they won't recognize the sugars as effectively as they would recognize a protein. So in the left-hand side here, you have a good example would be meningitis A, and you have a meningitis A polysaccharide. This polysaccharide would be directly recognized by B cells, and these B cells will produce plasma cells. So the important thing to consider here is this doesn't involve any T cell help. So you don't get any production of immunological memory if you are injecting a patient with just the sugar. So this is the B cell activation process independent of T cells, which we've said specifically only results in the production of plasma cells. On the right-hand side, this is where you have a conjugation of a polysaccharide and a protein. And basically, this would be the polysaccharide of the meningitis A, basically in conjunction with the tetanus toxoid, for example. And this would then basically drive more of an immune response. This would activate B cells in a T cell dependent manner. And this process would lead to the production of plasma cells, which can produce copious amounts of antibody, which is, of course, important, but it also leads to the production of memory B cells. And memory B cells, as we've said, are essential in providing immunological memory over the course of many years after you've received the vaccination. So if we move on and talk specifically about the next generation platforms for vaccine formulations, and specifically talk about the COVID vaccines. So these are the two types of COVID vaccines that are currently being administered and rolled out within the UK at the moment. So on the left-hand side, you have the Oxford AstraZeneca vaccine. On the right-hand side, you have the Pfizer BioNTech vaccine. So if we talk about the Oxford vaccine first, so this basically involves a chimpanzee adenovirus vaccine vector. So this is basically a virus that infects chimpanzees. Scientists have taken this virus, modified it in the laboratory, modified it to the point where it is unable to cause disease within humans. And they've basically used this virus as a vector for a SARS-CoV-2 spike protein. So they produced this vector and they've identified the genes that are encoded for the spike protein. So the spike protein is found on the surface of the coronavirus. So these are kind of like club-shaped spikes that are found in the outer coat of the capsid of the virus. They've basically identified the gene that's responsible for producing the protein, and via the process of gene sequencing and also genetic manipulation of the vector, they've basically inserted this gene segment into the vector of the adenovirus. And because this adenovirus is unable to cause disease, you are relatively fit and healthy after receiving the vaccine. And what happens once you've received the vaccine is that cells that will uptake the host cells that will uptake the vector will basically express the spike protein. They'll produce the spike protein, And these spike proteins will then be recognized by your antigen presenting cells, processed in a very similar manner to as we talked about earlier, and basically be presented to the adaptive immune system to drive immunological memory. And this immunological memory will result in antibodies specific for the spike protein. And therefore, at a later date, if you are infected with the COVID virus, then the immune system will recognize the spike proteins that are found on the surface of the virus. And therefore, you will hopefully remain fit and healthy. On the right-hand side, the Pfizer vaccine uses genetic material of the COVID virus and specifically uses the RNA of the COVID virus. So the RNA specifically would be isolated from the virus itself within the laboratory. The RNA will be encased within a lipid coat, so this prevents it from degrading. This lipid coat will also help the virus or help the RNA become inserted into the host cell. The host cell will integrate the messenger RNA, and the messenger RNA will basically instruct the cell to produce the viral proteins. So these viral proteins will then be processed by the antigen presenting cells, presented to the T cells to drive an immunological, to drive the adaptive immune system and provide and produce this immunological memory. So the last two things I want to talk about here are just some terms that you will likely encounter when thinking about vaccines, and if you're ever reading publications about vaccines. So we're going to talk about vaccine efficacy, so basically how effective a certain virus is, and also talk about the principles of herd immunity. So this is just another diagram here on the right-hand side, just to emphasize how effective vaccines are. This is from the US, basically taking into account the level of annual morbidity within the US. I think it's a few years old now, but it's still quite an important figure, given that the vast majority of these diseases have essentially been either abolished or severely reduced incidence within the US following the vaccination. So we should hopefully by now appreciate that vaccinations have substantial impact on the human population. It's averaged that about 6 million lives, if not more, are saved annually because of vaccinations. This second point here is quite an important one to emphasize how important vaccines are. So their role in reducing human mortality is second only to the provision of safe drinking water. And I think to me, that is quite an emphatic statement. But when we're talking about vaccine efficacy, specifically scientists can work this out by referring to the reduction of incidence of cases of a given disease amongst those that have been vaccinated. And this is all relative to the incidence of cases of the disease in the unvaccinated. And basically, this allows the scientists to work out how effective a vaccine is. And vast majority of cases, vaccines are 95% effective. So I don't think there's any accurate statistics for how effective COVID vaccines are. And I think this is simply because they've only recently been produced and we don't have any long-term studies assessing how effective they are. But there is, I think, some postulation that they may be anything from about 70 to 90% effective, the current two that are being administered in the UK. So it's just some more statistics just to emphasize how effective vaccines are. And vaccinations exist for most common infectious agents of the 20th century. So things like smallpox, polio, diphtheria, measles, mumps, and rubella. And as you can see from the percentage reductions, these are very, very good at doing their job. These vaccinations have reduced the amount of cases of a given disease by anything from about 98 to 100%. So the second thing I'd like to talk about is herd immunity. This is probably something you have have heard in the news or has been maybe mentioned by politicians in the last 6 to 12 months. But the idea and the principle of herd immunity is that not everyone can be vaccinated, okay? But you can still prevent the spread of disease via herd immunity. So herd immunity, the idea of herd immunity is that disease transmission can be interrupted with vaccination of the majority, but not everyone. And a great example is smallpox. So smallpox, as we've said earlier on in the session, was essentially the first disease that Edward Jenner vaccinated against by using the cowpox virus. But you need to be aware that not everyone received the smallpox vaccine when it was administered, when it was given worldwide. However, the transmission of this disease was interrupted simply because of this idea of vaccination, this idea of herd immunity. And this is essential, as I've said, is everyone cannot get vaccinated. And this next slide, hopefully it plays. If it doesn't, then I'm sorry. Oh, it does play. That's good. So what you'll see in this here is how herd immunity essentially works. So we'll work for each of them individually. So if you think about first population here, if none of the population is vaccinated, then of course the disease is can very easily transmit from individual to individual. Very similar case for 25% of the population being vaccinated, 50%, and even up to 75%. But you can see there's a drastic difference between the amount of people that have the disease. If 75% of the population is vaccinated compared to 50%. And then if we look at 90% vaccination or 95% vaccination, you can see from the yellow dots that these are all the people that are vaccinated and the blue dots are those that aren't. And you can clearly see that the disease is struggling to spread from individual to individual simply because the vast majority of the population is vaccinated. And this is essentially the principles of herd immunity. And scientists have worked out, very much dependent on how infectious a given microbe is, they've worked out what the herd immunity threshold would be for each given disease. So things like diphtheria would be 85% of the population would need to be vaccinated to reach herd immunity. Things like smallpox would be anything up from 83 to 85%, rubella, polio slightly lower. But the take-home message is it very much depends on how infectious the microbe is. And the more infectious the microbe is, then of course the threshold would be higher. For COVID, again, there's not enough statistics and not enough information for scientists to accurately determine what threshold this would be. But again, I think it's anything from about 90% to 95%. There's some suggestion in the literature that it could be anything from 90 to 95%, simply because it's a really infectious virus. So if we just want to finish off and talk about problems associated with creating vaccines, and specifically this idea of mutations within viruses. So it's important to consider that we've talked about other anomalies when it comes to infectious agents when creating vaccines. So things like fungi and parasites, it's quite difficult to create vaccines against these. Of course, you need to take into account the monetary costs involving creating vaccines. So of course, if you think about the two COVID vaccines that we've talked about, one of them has to be stored at -80 degrees before it's administered, and therefore the costs of creating these is a lot higher, and transporting these vaccines is a lot higher than the other one, which you can actually just store in the fridge. So of course, the obvious problems are present when it comes to creating vaccines, but there are other specific problems, and the specific problems is what we're going to talk about now in the context of mutations. So you should by now be aware that viruses have the potential to mutate, and the technical and scientific terms for these mutations are either antigenic drift or antigenic shift. So antigenic shift is what is essentially occurring within influenza A, antigenic drift is what is essentially occurring in the COVID variants that you have probably heard about in the news. And antigenic drift is where basically the proteins that are found on the surface of the vaccine are slightly different shaped after the mutation process. And obviously, if you have been vaccinated against the circular antigens, for example, these half-moon shaped proteins would therefore not be recognized by the immune system, and therefore you were essentially not vaccinated against these, and you would have an elevated immune response against these compared to these. Antigenic shift is where basically different types of viruses will reproduce and mutate to form a new strain. And what in the new strain, you'll have a combination of the different proteins that are found on the surface. And this is essentially what is happening in influenza A mutations from season to season. And this is essentially what is shown here. So as I've said, influenza viruses have the potential to mutate from season to season. And this, of course, reduces the effectiveness of viruses. You can see in this figure on the top right-hand side, the level or the effectiveness of a virus from, the effectiveness of the flu vaccine from season to season will, of course, be different depending on the level of mutation within that virus. So, for instance, in 2017 to 18, the winter of this season, of these 2 years, of course, clearly the level of mutation was quite excessive within the influenza A virus. And this is ultimately summarized here from what I've said. So we've talked about antigenic drift here, where you get a process of genetic change that leads to more variety within the proteins on the surface. Antigenic shift is where you make a new subtype, where basically there's reproduction between two strains that results in the production of a new subtype. And basically, a different way of looking at it, and specifically within influenza A, is these proteins here on the surface of the influenza A virus. And these two proteins are known as hemagglutinin and neuraminidase. And what you need to be aware of here is that there's 18 different types of H protein, so 18 different types of hemagglutinin protein, and 11 different types of the neuraminidase protein. And as I'm sure you can imagine, and it's simple mathematics, that the more variation you have in the shapes of the protein, and the combination of these will result in a vast number of different combinations. So you're going to have up to 144 different combinations of these H and N proteins on the surface of the influenza A virus. And that is simply the reason why from season to season, the vaccine will have to slightly change. And that is probably the biggest problem when talking about vaccines, and specifically talking about flu vaccines, is that from season to season, the flu virus will change the shape of the proteins on the cell surface. And therefore, this is why you will constantly need to have flu jabs from year to year. So that concludes the session, almost at an hour, which is good. So these are just some final remarks. The primary aim of vaccines are to enhance immunity against disease. You should be able to understand, at least to a basic level, the immunological mechanisms behind vaccinations. Excuse me. There's different types of vaccinations exist. So these can be comprised of whole organisms or essentially components of those infectious agents. I've introduced to you the idea of vaccination efficacy, and this is essential in trying to remember and trying to calculate how effective a certain vaccine is within a given population. And you should be able to recall the importance of herd immunity. And then the last thing I want to mention here, and this is something specifically for the oral science master students, but for the BDS3 cohort, this is something that you could potentially consider as well when you are watching this recorded lecture. For the master students, this is what we'll discuss in the live part of my session. So can vaccinations be, one, produced and two, delivered by dentists against different oral diseases? And is this possible? And if it is possible, you need to consider different things. So which microbe would you target? Would the vaccine be to prevent or treat the disease? When would you vaccinate and who do you vaccinate? And then, of course, there's immunological considerations as well. And the next slide shows 3 papers, which I want the master's students to go away and consider. I will make everything clear in the email that I send to everyone before the end of the, or before the session. But here are 3 papers to consider. So basically this is a potential vaccine against caries, and these 2 papers on the right-hand side are potential vaccines against periodontitis. So when you are reading these papers, I would try and consider each of these points. And as I've said, for the oral science master students, this is what we will discuss in the live part of the session for BDS3. Please feel free to go away and read these papers and also consider these points as well. But that brings us to a close for my vaccines lecture. If anyone does have any questions, specifically BDS3, if you have any questions, please feel free to email me. For the oral sciences cohort, I will try and take any questions in the first part of the live session. So if anyone does have any questions, feel free to write them down and ask me in the live part of the session. So thank you for your attention, and that is the end of the vaccines lecture.